You know those people, right? Someone in their 80s, maybe 90s, but their memory is just as sharp as ever. They totally defy our expectations of what aging is supposed to look like. Well, today we are going deep into the science behind these incredible minds. We're going to explore a groundbreaking new study that's starting to crack the molecular code for lifelong memory. Look, we all know aging affects our memory. For some people, it's a pretty gentle slope. For others, it can be a truly devastating disease. But then there's this small group where the mind just holds on. It seems to resist the pull of time, and for neuroscience, that has been one of the biggest mysteries out there. And that is the big question. This is what scientists are finally starting to figure out. And it's not just some curiosity, right? Answering this question really holds the key to understanding and, maybe one day preventing cognitive decline for every single one of us. So to get to the bottom of this, researchers looked at two complete extremes. On one side, you've got the SuperAgers. These are folks over 80 whose memory is as good as someone 30 years younger. And on the other side, of course, you have Alzheimer's disease. The thinking was, if we can compare these two groups, maybe we can find the actual secret to what makes a brain resilient. For the longest time, for about a century, the accepted wisdom in science was that you're born with all the brain cells you're ever going to get. That's it. But what if that was just wrong? What if the brain could actually repair itself, could replenish its own cells, even late in life? Well, there's a name for this idea: neurogenesis. It's the process of creating brand new neurons, and it happens in a really important area for memory, a place called the hippocampus. And let me tell you, this one idea completely flips the script on how we think about the aging brain. And this breaks it down perfectly. It all starts with a neural stem cell. Think of it like a seed. That seed then gives rise to something called a neuroblast, which is kind of like a little sprout. And then that sprout grows into an immature neuron, a sapling, that will eventually get wired into the brain's network. The ability to finish this journey turns out to be everything. So, okay, that's the theory, but how on earth could scientists actually see this happening inside a real human brain? They needed a totally new kind of map, one that could chart the inner world of individual brain cells with just an incredible level of detail. And the scale of this thing was just massive. We're talking about researchers analyzing the genetic machinery of more than 355,000 individual cells taken from the brains of SuperAgers, healthy older adults, and people with Alzheimer's. This is truly big data, but for the human brain. So, all of that data creates this. Now, I know it looks complicated, but just think of it as a super detailed atlas of the brain's memory center. Every one of those colored dots is a different type of cell, which let the scientists see exactly what was going on with the stem cells, the sprouts, and the saplings in every single person. And buried inside that atlas, they found an absolutely stunning clue. The biggest difference between SuperAgers and those with Alzheimer's, it wasn't just about the genes they had. It was about which genes their cells could actually get to. Imagine your DNA as a giant library full of instruction manuals. The key isn't just having the manuals, it's about which ones are unlocked and available to read. The study, of course, puts it in very sciencey terms. It says the problem was chromatin accessibility. But all that means is how tightly your DNA is wound up. If it's coiled up too tightly, the cell can't read the genes it needs to build new neurons and repair itself. That all-important instruction manual is effectively locked away. And this discovery was the key. It allowed researchers to finally decode what makes a SuperAger's brain so incredibly special. They found a distinct molecular blueprint, a signature for resilience. And the differences they found were not subtle. Compared to their peers, SuperAgers had a 2.5 times increase in the number of new immature neurons. They had way more of those sprout cells. And their cells had this unique resilience signature. They were turning up the volume on genes that are critical for brain health, like one called BDNF. So here's the bottom line. SuperAgers aren't just getting lucky. Their brains are actively working to maintain this youthful, regenerative environment. It is a fundamental molecular advantage that is keeping their memory sharp. All right, so that's the story of resilience. But now we have to look at the other side of the coin. What happens when this whole process of creating new neurons just breaks down? This is where the story reveals the molecular tragedy of Alzheimer's disease. This chart says it all, doesn't it? Neurogenesis is sky high in SuperAgers. It's chugging along okay in healthy aging, but it just drops off a cliff in Alzheimer's.
the brain's ability to generate new neurons is severely damaged. The factory is basically shutting down.
And this image shows us why. What you're looking at is basically a genetic fingerprint. In the brains of people with Alzheimer's, those key genes for making new neurons and building connections, they become inaccessible. That instruction manual we talked about, it gets locked up tight, and the brain can't perform its own repairs.
Now, here's the really crucial part. This isn't just a symptom that shows up at the end. This study found that this whole process of locking away genes, it starts early. We're talking about the preclinical stages, before a person even shows major signs of memory loss. It's like an early warning signal written in the code of our cells.
Okay, so we have the resilient signature of the SuperAgers, and we have the failure signature of Alzheimer's. So what does this all mean for the future? What can we do with this new information? It means that for the very first time, we can see that having an exceptional memory in old age isn't just some random quirk of fate. It is a real biological phenomenon. It's a specific molecular signature that promotes the birth of new neurons. And because we can now identify it, we can study it, we can learn from it, and maybe one day we can even replicate it. And that leaves us with this final amazing question. By fully decoding the advantages of the SuperAger brain, could we develop new ways to protect our own minds? Could we find a way to keep that instruction manual for brain repair open and accessible, and unlock the secret to lifelong mental clarity for everyone? It feels like the map is finally in our hands.